Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about the cellular respiration. It is important for plus 2 first year student, plus 3 student, M.Sc student as well as SSB, TGT, PGT and lectureship aspirants. Cellular respiration is defined as a process of oxidation of complex organic molecule into simpler inorganic molecule or simpler inorganic substances like carbon dioxide, water and energy in the form of adenosine triphosphate or ATP. Simply, respiration is the oxidation of organic fuels or any organic molecules, commonly the glucose, by molecular oxygen. Respiration is the process where oxidation of organic fuel occurs by the molecular oxygen. This process can be divided into two categories. One is aerobic respiration, second one is anaerobic respiration. Aerobic respiration occur in the presence of oxygen and anaerobic respiration occur in the absence of oxygen. In aerobic respiration, after glycolysis, Krebs cycle occur, and after Krebs cycle, electron transport chain and oxidative phosphorylation takes place. Glycolysis is a metabolic reaction where glucose break down to form the pyruvate or pyruvic acid. It is a 10-step reaction which takes place in the cytoplasm of the cell. This process is common to both aerobic and anaerobic reaction. It is a 10-step reaction where the first step is glucose is converted to glucose 6-phosphate. Then from glucose 6-phosphate, fructose 6-phosphate is formed. Then from fructose 6-phosphate, fructose 1,6-bisphosphate is formed. Then fructose 1,6-bisphosphate splits to form dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate, from glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate is formed. Then from 1,3-bisphosphoglycerate, 3-phosphoglycerate is formed. Then 2-phosphoglycerate, then phosphoenol pyruvate, and from phosphoenol pyruvate, pyruvate is formed. Anaerobic respiration can be of two types. One is ethyl alcohol fermentation and lactic acid fermentation. So in case of ethyl alcohol fermentation, first glucose is converted to pyruvic acid in the process of glycolysis, and glycolysis is common. The glycolysis pathway is common to both aerobic and anaerobic respiration. At first 2-pyruvic acid is formed. Then from pyruvic acid, 2-acetaldehyde with 2-carbon dioxide formed by the presence of pyruvate decarboxylase. Then from acetaldehyde, by the presence of alcohol dehydrogenase, 2-ethyl alcohol formed. Then in case of lactic acid fermentation, which takes place in certain bacteria like lactobacilli and streptococci and muscles tissue of animal, the end product is lactic acid. This process is catalyzed by lactate dehydrogenase. So at first the glycolysis producing 2-pyruvic acid. From pyruvic acid, using lactate dehydrogenase enzyme, 2-lactic acid will be formed in the process of lactic acid fermentation. So after the formation of pyruvic acid, then acetyl coenzyme A is formed. Under aerobic condition, pyruvate produces the acetyl coenzyme A in the mitochondrial matrix, and then acetyl coenzyme A enters into the Krebs cycle to carry out the aerobic respiration. Formation of acetyl coenzyme A occur by oxidative decarboxylation. What is oxidative decarboxylation of pyruvate? Oxidative decarboxylation means carbon dioxide is removed in the presence of oxygen, and this reaction is catalyzed by pyruvate dehydrogenase complex. So the whole reaction is pyruvic acid, 1 coenzyme A is added and 1 NAD plus. In the presence of pyruvate dehydrogenase complex, acetyl coenzyme A is formed and 1 carbon dioxide released, because pyruvic acid is 3 carbon compound and acetyl coenzyme A is a 2 carbon compound, and finally NADH is produced. So acetyl coenzyme formed from pyruvate also act as a key intermediate in the formation of fatty acids and biosynthesis of lipids. So it is the link reaction between glycolysis and Krebs cycle. Then after the formation of acetyl coenzyme A by the oxidative decarboxylation, acetyl coenzyme enter into the Krebs cycle or tricarboxylic acid cycle. It is called as Krebs cycle because this cycle was discovered by Sir Hans Krebs. So according to his name, this cycle named as Krebs cycle. So acetyl coenzyme A is a 2-carbon compound enter into the Krebs cycle. Then oxaloacetate by adding with the oxaloacetic acid, the acetyl coenzyme A form the citrate. Acetyl coenzyme A is a 2-carbon compound, oxaloacetate is a 4-carbon compound. So togetherly they form a citrate with 6 carbon. Then one unstable compound, that is 6-cis aconitic acid is formed. Then by the presence of aconitase enzyme, isocitrate is formed. Then here after isocitrate, oxalosuccinate, another unstable compound is formed, and one NADH also removed and released during this reaction. Then finally alpha ketoglutarate is formed from oxalosuccinate. Oxalosuccinate is a 6-carbon compound, but this alpha ketoglutarate is 5-carbon compound, so one carbon dioxide is removed. Then from alpha ketoglutarate, succinyl coenzyme A is formed, which is a 4-carbon compound, so here also one carbon dioxide is removed and one NADH is formed. So till now 2 NADH formed, one is here and one is here. Then from succinyl coenzyme A, succinate is formed and one GTP actually, which is equal to ATP is formed, and finally forming succinate. From succinate, fumarate is formed, and from by releasing one FADH2. Then from fumarate, malate is formed. Then from malate again oxaloacetate is formed by removing one NADH. So at the end of the cycle, 3 NADH, we have 3 NADH, 1 FADH2 and 1 GTP, which is equal to ATP. So it is the complete Krebs cycle. After the Krebs cycle, mitochondrial electron transport chain and oxidative phosphorylation occur. So mitochondrial electron transport chain or terminal oxidation is the last phase of aerobic respiration. During this phase, NADH and FADH2 released from different reaction stages of cellular respiration are oxidized by transferring their electron to terminal acceptor oxygen or O2 through a chain of electron carriers. Finally, oxygen is reduced to form H2O. So this mechanism of oxidation of reduced coenzyme through electron transport chain is associated with phosphorylation of ADP to ATP. Thus the process of ATP synthesis also coupled with electron transport chain called oxidative phosphorylation. So this in case of eukaryotes, all the components of this electron transport chain are embedded in inner mitochondrial membrane and its cristae. They bear numerous elementary particle or F1, F0, F1 particle. That F1 particle is actually the ATP synthetase. But in case of prokaryotes, the components or the enzymes for electron transport chain are located in the plasma membrane, as they devoid the mitochondria. They lack mitochondria. So the enzymes of electron transport chain in prokaryotes are located on the plasma membrane. So these electron transport chain enzymes are arranged forming 4 complexes on the inner mitochondrial membrane. So these are the complexes. It is the complex 1, then complex 2, complex 3, complex 4, and finally it is the ATP synthase or ATP synthetase complex or F0, F1 particle. F1 particle is the ATP synthase enzyme through which the ATP is formed. So electron carriers are accomplished by transfer of electrons from donor to acceptor. So here one donates the electron, another accepts the electron. Such oxidation reduction reaction which occur simultaneously in this cycle are called redox reactions, and they are catalyzed by a variety of dehydrogenase, reductase, oxidase. So the most accepted sequence of this electron carrier are first NAD, then FAD, then coenzyme OQ, then cytochrome B, then cytochrome C1, then cytochrome C, then cytochrome A, then A3, and finally O2 accept this electron forming H2O or water. So this is the sequence of electron transfer. So at first complex 1, which is NADH COQ reductase or simply NADH dehydrogenase. So at first in complex 1, the NADH bind, NADH plus H plus bind, and then transfer its electron to FMN, flavin mononucleotide. Then FMN accepts that electron and transfer the electron to FMN. Then from NADH, FMN accept the proton and reduce to FMNH2. Then FMN pass it to the FES protein complex. In this process, total 4 protons are transferred from the matrix to the intermembranal space. And finally, the electron is transferred from FMN to FES, then from FES to other complexes. Then complex 2, which is succinate COQ reductase, it consists of FAD and FES protein. So here FAD is reduced to FADH2 by accepting H2 from the succinate. It becomes FADH2 by accepting the hydrogen atom from the succinate, and it becomes FADH2. Then it transfers 2 electrons to FES protein, and then it moves to COQ or ubiquinone, which is a mobile carrier, and COQ accept the proton forming COQ H2 and then 2 electrons. And here also in this process, proton is transferred from matrix to the intermembranal space. Then complex 3, which is COQ cytochrome C oxidase. So cytochrome C oxidase or oxidoreductase, it contains cytochrome B and cytochrome C1, and some FES proteins are there. So electrons also transfer from that complex, and in that process also 4 protons are transferred from matrix to the intermembranal space. Then complex 4, where cytochrome A and A3 are present, they accept the electron. Then finally the electron move to the half O2. Then by accepting electron and 2 proton from the matrix, the half O2 is converted to H2O. In this process, only 2 protons move from matrix to the intermembranal space, because 2 protons are already utilized in the formation of water. So in this process, many protons are moved from matrix to the intermembranal space. So it creates a gradient, because there are more proton in the intermembranal space than the matrix. Then that protons move into the matrix. They try to move into the matrix to balance that gradient or to create a equilibrium. So during their movement from intermembranal space to the matrix, they flow back through ATP synthase or F0 F1 particle. During their movement, ADP plus inorganic phosphate form the ATP. So in this way, with electron transport chain, oxidative phosphorylation is accompanied, and finally ATP is formed inside the mitochondrial matrix. So it is the overall electron transport chain and oxidative phosphorylation. And it is the complete your it is your complete aerobic respiration. I have already told you about the anaerobic respiration, and here it ends the aerobic respiration. So it is all about respiration. I hope you really get benefit from it, enjoyed this tutorial session, and in my next video I will upload other important topic of biology. So please stay tuned and enjoy my videos like this, and please like, share and subscribe to my channel Sunanda's Tutorial. Thank you.